Hi everyone. This PowerPoint is just meant to review protein synthesis. Have your notes and vocabulary handy to follow along. If something is still confusing or you have a question, write it down and then send it to your teacher. Just a reminder, please make sure you are taking notes. They will help you in learning these concepts and come in handy when answering the questions for an eventual quiz. Alright, let's jump in. Imagine you are at main event and you want to play air hockey. The only problem is that the air hockey table only accepts game tokens, and all you have is a $5 bill. What do? Do you go home in defeat? No. You can change your cash for game tokens at a token machine. In a similar way, your cells cannot make proteins directly from DNA. They must convert DNA into a middleman, a molecule called RNA. Then, from the genetic information stored on an RNA molecule, protein can be made. This is the process of protein synthesis. Without the RNA molecule, specifically the messenger RNA, DNA cannot code for proteins. It needs that middleman. Additionally, you might remember that ribosomes are the protein factories in the cell. Well, where is the DNA? In a eukaryotic cell, it's in the nucleus. Can the DNA leave the nucleus? No, it's trapped. So, what does it need to get its message out? It needs a messenger, in this case, messenger RNA. So today we are going to review this process, the central dogma or the main idea of how DNA codes to make proteins. Just like how at main event, the money is transcribed into the tokens to be able to play the game. Just like how at main event, the central dogma involves three main steps. First, DNA is replicated. We learned all about this when we learned about the structure of DNA. DNA is then transcribed into RNA. Both are nucleic acids. They speak the same language. Then, the mRNA is translated into a new language of amino acids to make the protein in the last step.
So step one of the central dogma is DNA replication. You remember DNA replication, right? Helicase, DNA polymerase, etc. Here's a visual that shows the process of DNA replication. Remember, we start with one DNA, and then we end up with two identical copies of DNA. Remember, the end result of DNA replication is two identical DNA molecules. Each new molecule is made up half of the original DNA and half of the new DNA. Do you remember when DNA replication happens? I know it's been a while. Replication occurs right before mitosis or cell division, specifically in interphase during the synthesis part, the S phase. The new cell needs a copy of DNA during mitosis. Remember, each daughter cell needs a full set of DNA. Where is DNA located again? Oh yeah, that's right, it's in the nucleus. Now, can DNA ever leave the nucleus? No, you're right. It always stays in the nucleus. It's too valuable to leave the nucleus. So replication always takes place in the nucleus of the cell because that's where DNA is located. So we learned the different steps of DNA replication before. Remember, the DNA is unzipped by the enzyme helicase. Then, the DNA acts as a template to create the complementary strands. The enzyme DNA polymerase is the master builder that puts the new bases together. Apples in the tree, cars in the garage. The end result of DNA replication was two identical DNA strands. Each of the new strands is made up half of the original DNA and half of the new DNA. You can see this in the picture on the slide, where two identical DNAs are each half purple for the original strand and half green for the new strand. Here's a short video to review DNA replication. Enjoy, and don't forget Mr. Stefanov misses you very, very much.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
First step of Essential Dogma created identical DNA strands. In this section, you will learn about the second step, transcription. Transcription is the process of copying, or you can say transcribing, a sequence of DNA to produce a complementary strand of RNA. The RNA that is produced is like a temporary copy of DNA that leaves the nucleus in order to make protein. It is like a messenger, and that is why we call this RNA molecule messenger RNA. Remember that DNA is stored permanently in the nucleus, and transcription is the copying of this DNA. So this means that the process of transcription takes place inside the nucleus. Other things we need to know about transcription are the 3 steps, and there are 3 different types of RNA that are produced. The process of transcription is much like DNA replication. In the first step, DNA is unzipped to form 2 separate strands, and it uses the same enzyme helicase. In step 2, RNA nucleotides are going to form complementary base pairs with one side of the DNA. Remember that complementary base pairs simply means A's pair up with T's and C's pair up with G's. In the third step, the new RNA strand detaches and leaves the DNA molecule. This is a visual representation of transcription. So in the first step, this is when the DNA is unzipped by a helicase. In step 2, this RNA polymerase in green is going to match nucleotides to the original DNA sequence, creating a long chain of messenger RNA. And then in the third step, new RNA strand is formed, and it's going to detach from the original DNA molecule, and then everything is zipped back up into its original state. Now, last week we learned about the difference between DNA and RNA, and one of the differences is that instead of thymine, which is the T in DNA, RNA has a uracil, which is coded as U. So you can see on the right picture, the above strand is the original DNA sequence. So we see thymine in the second and the fifth nucleotides. Okay. And the green down below is our mRNA. And you notice that the U is pairing up with the A this time. Normally we would think T's pair up with A's, but in this case, RNAs, they do not have thymine, instead they have uracil. We're going to do some practice problems with you. So imagine this following DNA sequence. We have TCG, ATA, and ACG. And we want to know what is the complementary RNA sequence. So I'll give a pause for you guys to think about, but remember our base pairing rule, and you can see that in the right hand corner. Okay, so you should have gotten ACG, UAU, UGC. Here's another example. This DNA strand is ACG, AAT, GGC. What is a complementary RNA? You should have answered UGC, UUA, CCG. Remember is that there are 3 types of RNA necessary for transcription. There is mRNA, which is the messenger molecule that takes a message from the DNA in the nucleus to the ribosome. There is ribosomal RNA, which is rRNA, which actually makes up part of the structure of ribosomes. And then there is tRNA, which transfers the amino acids during translation, step 3 of essential dogma.
All right, so the last step of the central dogma, or also known as protein synthesis, is translation. So what happens after transcription? Well, imagine that the DNA and the RNA speak the same language, but the protein speaks a different language. What do they need to communicate together? They need a translator. RNA and DNA speak the same language. That language is the language of nucleotides. The A, T, C, G, and U. Proteins speak the language of amino acids. Therefore, the instructions that RNA has needs to be translated. That's why step 3 is called translation. It's the process that converts or translates an RNA message into a protein. This all happens or occurs in the ribosomes, the organelles inside of the cell. The first step of translation, after transcription has already happened, and we've changed from DNA to RNA, the messenger RNA, called mRNA for short, is a molecule, and it's going to leave the nucleus. Okay, it's very important. It's going to come out of the nucleus into the cytoplasm and go to the ribosome. Okay, this mRNA carries the genetic code that has already been transcribed from the DNA. The second step of translation is the mRNA is going to travel to the ribosome, okay, and then it's going to be joined by tRNA molecule, which is just another type of RNA. They literally named it tRNA because it looks like a T. Okay, the tRNA is important because it carries the protein ingredients, which are called amino acids. Step 3, the last step of translation, the ribosome and the tRNA are going to work as a team to put the amino acid parts together to form a protein. Looking closely at translation, it's important to understand what codons are. Codons are 3 mRNA bases in a specific order that code for an amino acid. So here, circled in red, there's a codon AUG, A-U-G, and it codes for the amino acid methionine, but to abbreviate it for short, we put MET. Okay, so on the mRNA, we have the codon AUG, AUG, and you see the tRNA, and it's going to bring the MET amino acid, or the methionine. AUG is important to remember because it signals the start of a polypeptide chain. So we know, oh, whenever you see AUG, okay, an amino acid chain is going to start growing here. An easy way to remember this is that AUG is an abbreviation for August, and that's when school starts. And AUG is the same, that's when an amino acid, or it's going to start building a protein. As for RNA, or tRNA, this molecule is going to carry and bring the matching amino acids to the ribosome for each different codon. Okay, tRNA, we look at this little guy, he has 3 teeth. They're not teeth, those are the bases, okay? The 3 bases in a row right there, we're going to call them anticodons. Okay, because they are the they are the opposite, they're going to bind to the mRNA. If you think about it, codons are a set of 3 bases in a specific order that code for a specific amino acid. So each tRNA is going to be different because they have their own code that they are responsible for matching up. So the AUG is going to bind to a specific tRNA that would be UAC. The tRNA would have a uracil. The first base would be uracil, the second base would be adenine, and the third base would be cytosine, so that it can match up to the AUG codon, bringing the methionine with it. Today, you'll be looking at how to use a codon chart like this to be able to code, or rather decode, mRNA into amino acids. So for example, if we had OG, okay, AUG, A comes first. So on the left-hand side, there's an A. Okay, we're going to look at just that row of A. All right, the second letter is U. So I'm going to look at the top and find which row or column is U, and it's the first column. So now I know that it has to be in this box from the left, not the very bottom, but the one right above it. Okay. And then the last is G, OG. And then just by looking at it, which one is AUG? AUG right there at the bottom equals met, or methionine.
Hopefully that review helped. If you have questions, make sure to reach out to us. That's what we're here for. We miss you all. Next, you're going to watch a Khan Academy video to just review this process one more time.
So what exactly is the central dogma of molecular biology? Well, really it could just be called the central dogma of all of life, because it explains how you and I take this conglomeration of genetic information from each of our parents and how this information gets transferred into generating a full-blown human being like you and me. So some very clever scientists, Francis Crick and James Watson, or Watson and Crick, as they're often referred to as, are credited with discovering this dogma, which they say deals with the detailed residue-by-residue transfer of sequential information. Or, as Marshall Nierenberg, who won the Nobel Prize in Physiology and Medicine, once said, DNA makes RNA makes protein. And I think this simple explanation really just says it all. So let's explore this concept a little bit further. So we have three major players here. DNA and RNA are nucleic acids, which are made up of nucleotides. And proteins are made up of amino acids. And the information starts at the most basic level stored as DNA, which can then be restored as DNA when DNA copies itself in a process called replication. Then DNA can be copied into RNA in a process called transcription. And then finally you can use the information in RNA to synthesize a protein in a process called translation. Now, since DNA, RNA, and protein are linear polymers, this means that each individual unit or monomer is only attached to at most two other units. So say we have a monomer, which is just one unit. They are connected in a series like this, which makes it a linear polymer. And this is the same for DNA, if each of these is a deoxyribonucleic acid, for RNA, if it's a ribonucleic acid, or a protein, which are just amino acids all connected in a linear polymer. So what does this mean? This means that the specific sequence of each of these monomers effectively encodes information, and that the transfer of information is faithfully preserved from DNA to RNA to protein. Each polymer sequence is used as a template for the synthesis of the next polymer. And you could go into any step in this sequence and determine what the corresponding polymers would look like. So in other words, you could take DNA and obviously figure out what the corresponding RNA would look like, and then what the corresponding protein would look like. So this whole process is the central dogma. It can sometimes be a little bit tricky to keep all of these terms straight, so I'll try to break it down a little bit for how I like to remember them. For DNA, I think it's pretty easy. When you go from DNA and DNA makes a copy of itself, it's called replication because DNA is just replicating itself. It's making the same copy of itself. Transcription and translation, on the other hand, it's kind of easy to get these two terms mixed up. One of them obviously is talking about DNA to RNA, whereas the other one is talking about going from RNA to protein. So if you look at the word transcription, it has the word script in it. So I think of it as going from one written form to another kind of written form. And both use nucleic acid. So they both use this sort of alphabet, if you will, of nucleic acids. And so you're just going from one kind of alphabet to the next kind of alphabet. Translation, on the other hand, which is also the same term that we use when translating one language to another, describes going from nucleic acid to amino acid. So it's like you're using one kind of language and going to another kind of language, because you're going from nucleic acid building blocks to amino acid building blocks. So hopefully that helps you keep these terms straight a little bit. So what did we learn about the central dogma? Just remember the simple statement that DNA makes RNA